Yes, and then we have another scientist, Pernette Verschure. You come over. You're also an associate professor in cell biology and synthetic and systems biology. Yeah, I have to read it from my card.
Good.
And I think you also have something very interesting to tell about epigenetics.
Yeah, that's true.
I don't know exactly what they are, but I'm sure it will be a lot more clear afterwards.
Yeah, good. Is there somewhere
Yeah, I see one over here.
Yeah, it's this one. Okay. Good.
The floor is yours.
Am I set up?
Oh, just a sec. Maybe you can tell a little bit what epigenetics are.
Yeah.
Oh,
okay.
I think you can.
Yeah, good. So, somewhere in time, I want to talk about epigenetics, the time clocks, the epigenetic time clocks in our body. And these clocks are ticking away our time, so making us change from maybe a healthy condition to a diseased condition. So I want you to realize for a few seconds how you are today and how you were a year ago. And also how similar we actually are as human beings, but yet so different. So did you ever wonder why you are so different from your neighbor, your brother, your sister? And why some of us obtain a disease and others not? All these questions are very important. Now, I guess you all know our genetic material, the DNA, our genes, encode for the way how we are, our visual characteristics. And mutations in our DNA can cause diseases. Now, the good news is there is an additional layer of regulation, our epigenetic clocks, that actually control the way our genome is functioning. Now, the bad news is this layer is very complex, and we know still so few about this regulation in individuals. Now, for today, I want to make a short journey through the way our genome is functioning at the molecular level, the nanoscale level. So fasten your seatbelts, because here we go. Okay, our bodies, we have organs, for instance, like the liver, to detoxify. And the liver consists of tissues. And these tissues consist of cells. And within a cell lies the nucleus. The nucleus is the harbor of our genetic material. There is the DNA. And on the DNA are the genes. And these genes can be transcribed into RNA and then translated into proteins. And these proteins are the working horses in our body. They're very important. Now, I want to give you 3 puzzling issues regarding the functioning of our genome. The first one, monozygotic twins. They stem from one fertilized egg, so they have identical DNA. So when they're born, they're indeed very identical. But over time, when they age, they increasingly become more different. How is this possible? They still have the same DNA. Our body consists of many different cell types. We have approximately 250 different cell types. Now, all these different cell types have their own identity. Our skin cells, our brain cells, our liver cells. So each cell uses only a small fraction of the available genome. But each cell has the whole genome, all the DNA. How is this possible? Now, as I said, the DNA, if we would take it out of the cell nucleus and we would stretch it up, it would be a fiber of 2 meters. And it has to fit in this small nucleus of only 10 micrometer. So imagine a long wire, like 20 kilometers from Amsterdam to the beach, that has to fit in a tennis ball of only 10 centimeter. And then on that long wire, a gene would be 2 centimeter. So how can a cell find that gene and turn it on or turn it off in that wired rope in that small nucleus? How is it possible? Now, the answer is there is an additional layer of regulation, the epigenetic regulation. So epi comes from the Greek and it stands for supplementary, on top of. So this is what we call the epigenetic clock. Now what is this? Actually, it consists of chemical subgroups. So an acetyl group, a methyl group, a phospho group that are attached to the DNA or to the proteins that fold the DNA in such a way that it fits in the cell nucleus. Now, there is a whole layer of proteins that can either write these chemical subgroups, or they can erase them, or they can read them. And they can make the genetic material folded in such a way that it fits in the cell nucleus, and that some genes can be used and others not. So apparently, during embryonic development, this epigenetic layer is turned on, so that each cell actually uses only a small fraction of the genes, and another cell uses another fraction of the genes. So that's interesting. Now, because this layer is driven by proteins, and they can come off and be turned off, and they can be changed by the environment, this epigenetic layer is quite reversible. That's an interesting point, because actually, when we live and when we age, we're exposed to stress, to food, to environmental conditions, and our epigenetic layer can change our epigenetic clock. Now, I want to show you two examples illustrating that indeed environment can change this epigenetic clock behavior, but also that these epigenetic layers are actually transmitted, herited from one generation to the next generation. Now, children born from mothers that were exposed to famine during the Second World War, their epigenetic composition is remarkably different. Now, these children also have a higher risk for obtaining certain diseases, cardiovascular diseases, obesity, diabetes. And the intriguing point is that their children and grandchildren also show this changed epigenetic composition. Now, rats exposed to stress when they're having little pups show that they behave more different to their children. They show less licking and grooming. And these pups, their epigenetic material is changed. Now, intriguingly, these pups also treat their pups differently. And this change in the epigenetic layer is inherited through the generations. So these examples clearly show that the epigenetic layer is flexible and it can be changed due to the environment. Now, we find this topic very interesting. And in my research, we are mostly interested in how is this epigenetic clock switching? How fast? Which mechanisms? And can we actually predict then why people turn from a healthy condition to a diseased condition? Now, obviously, this is very difficult to study. So we start using computational models where we can actually play around with the epigenetic composition. So we can turn on some things that we know and then simulate it in a computer. And we can test it back in cells that we engineer in such a way that we can also drive that computer model within a cell. Now, I want to show you two examples where we simulated the epigenetic layer. In the first case, where we only recruit such a protein that drives a chemical subgroup, in this case the red signal, such that it binds on the genetic material. And in the second case, where we say, now, okay, this fiber is actually not an inert fiber, but it can also fold. And then at the positions where it comes close together, the epigenetic codes can actually switch. Now, if we simulate that, and I plot now here on the y-axis the genetic fiber, and on the x-axis we see the time. So in the first mechanism where we only allow the proteins to target on the genetic material, what do we see there? We see amazingly that there is like a separation of the one state and the other state. So the green versus the red, so the two different chemical subgroups show now a boundary. And then when we allow the material to fold, then you can clearly see that we lose this boundary. And now the chromatin or the epigenetic material can switch from the one state, so the green, to the red. So we find this very interesting because it shows that actually the epigenetic composition can switch very fast. So the hardware of the epigenetics can change at second time scale. Now, another thing we are very interested in is actually the differences between cells as a way to say something about differences between individuals. So we measure at molecular level the RNA levels in individual cells. So how do we do this? We use complementary pieces of DNA that are labeled with a tag that we can visualize in the microscope. And then we use advanced microscopy, confocal microscopy we call it, where we can make optical sections through our cells and then analyze in 3D the transcripts in individual cells. Now, how does that look like? In red, you can see the RNA, in blue, the DNA, so the nucleus, and in green, the contours of the cell. So this is a very robust way to actually detect in single cells, in individual cells, at molecular level, these RNAs. Now, where do we use this all for? We're also interested to use this in medical cases, so in biomedical applications. Now, in September, I got the very good news that a project that I had been writing, a huge project, an international project, was very well received by a European committee and would receive funding, 2.8 million, for 15 international partners to actually perform research for 12 PhD students. So when I told this very good news to my son, 8 years old, he immediately said, "Oh, Mama, that's a lot of money. You should buy a Bugatti Veyron." And then I realized I had to explain him very well that this amount of money is very useful for scientific research and much more important than spending it on materialistic items. So I realized I had to explain him carefully and to convince him that this research is truly important. So I told him, many women in the world obtain breast cancer. Now, some types of breast cancer are very well treatable and is known for long endocrine treatment. But after a while, when time passes by, some patients become resistant to the treatment. And then these patients are not well treatable anymore. Now we want to understand from the epigenetic layer whether we can actually predict why these patients become so resistant. So we want to screen their epigenetic composition in the sera, in the tissue, in the cells, and then plot it also on existing information using computational models and bioinformatics tools, and then test our observations by actually changing the epigenetic composition with novel epigenetic interference technologies, and then come up with an epi-predict model, so a prediction why these patients become resistant. Now, the take-home of what I want to tell you today is that in our body there are these epigenetic clocks, and they tick away our time, and they control the way our genome is functioning. Now, we are mostly interested in the switching, so in turning the buttons that make this epigenetic layer switch to understand why you go from a healthy condition to a diseased condition. And I think the challenge for the future is to understand these epigenetic clocks and also their timing in individuals, and to understand why one person is different from the other, to come in the end with treatments also based on individuals. So thank you for your information.
Yeah, thank you, Pernette Verschure. I found this quite hopeful.
Okay.
Because if I understood it correctly, we can change our genetics.
Well, that's maybe not the right way I would say it, yeah.
Okay.
But it's true that there is more than only genetics. And of course, yeah, people have tried with gene therapy to treat, and that's very difficult to change your DNA, obviously. Mm-hmm. But there are now ways to actually change your epigenetic composition, and because it's so reversible.
But could we, like, for example, also fight aging, which I'm not. I don't care about aging yet, but probably in a few years. Is that
Yeah,
possible?
I think this is, of course, also one of the topics that is related to this, that actually from the epigenetic material, we can already read the age of a person. Actually, in a forensic project, there was one of our speakers was in the forensic field. What we're now going to do is to see whether, based on the epigenetic layer, we can actually predict, like a spot on the crime scene, what would be the age of the person.
Okay.
So there is really information also about the age.
Okay, well, interesting. Let's give another applause for Pernette Verschure.